Introduction. Sharon, you have the floor.
Good job. So good morning. My name is Sharon Nachman. I'm the Division Chief for Pediatric Infectious Disease at Stony Brook. I am a distinguished professor and an endowed chair at the institution. I'm also the PI of the maternal child HIV network called IMPAACT. So I have no conflict of interest. I don't get any pharma money. So that's always very nice. My husband's still waiting for that. But today, I'm going to talk about AI in clinical trials. And I thought I would spend some time talking about what clinical trials are, what they mean, what we're going to talk about, and then some of the uses that we're putting in our EMR, looking at clinical trial sort of data sets, and then where we're moving AI into in real life in clinical trials. So sort of a big range of topics. I know I have a long time to talk, but I'm hoping that, if you have a question on something I'm talking about, let's stop there, because, I have 60 some odd slides. By the time I get to slide 60, you're like, well, what did she say in slide 32? So that's not going to be very good. So I'm excited to be here today. So first of all, with clinical trials, people always say, so how did you get here? And the answer is a lot of time. If you're an adult person and you're doing adult medicine, the FDA was very kind to you. And they said, good, here's the rule. You go ahead and enroll adults on a clinical trial. Fine, you're done. When it comes to us pediatricians, they're like, oh, no, stop. You cannot do trials in children. Oh, you want to do trials in pregnant women? Oh, my God, let me lay in the street and get run over by a car. Of course we cannot do trials in pregnant women. And that has resulted in 10+ years delay in any new medicine ever coming to children and never coming to pregnant women. And that, as you can imagine, is a big problem if you're a pediatrician or a maternal child specialist. We actually have no licensed data in children. So I thought I would start with, how do we actually get to do a clinical trial? Well, the drug is developed, and I'll just skim right over that. You were in the lab, you developed a drug, then you tested some animals. What's the problem? If you tested adult animals, that was really nice. But if you didn't test infant animals or pregnant animals, you can never move your drug to be tested in children or in pregnant women because you lack the data. So the FDA a couple of years ago went, oh, aha, that's a problem. So now every company is required to test their drug in 2 different animal models. You're going to ask me, so great, does that happen? No, it does not happen. Oftentimes, they will run an adult animal, submit to the FDA, do an adult study, and then if the adult study is successful, and we say, well, what about children, pregnant women? They will then turn around and go, oh, right, I have to run this in pregnant animals or in infant animals. That puts another 3+ years delay until any time you'll actually ever get to look at that drug in children or pregnant women. What's the other problem? The animal models that are picked don't necessarily mimic childhood or pregnancy. And if you can imagine, pregnancy is 9 months in an animal, a couple of weeks, maybe a couple of months. And in pediatrics, that first year of life seems like 10 years. After that, it seems like another 100 years till they finally go to school. In an animal model, it's 3 weeks. So there's never enough time in the animal models that are available to actually predict any kind of safety. You could get the big adverse event, the animal died, but you won't get anything small of they had a headache or their arm hurt or their leg hurt. All right, so then we have the animal studies are done. Then they have an IND application. They submit to the FDA and say, we'd like to test this drug in humans. Then they do a phase 1 study. A phase 1 basically is picking a dose. And the doses can range from any time from 1 times the dose to 1,000 times the dose. And because pharma companies would like to save money, they kind of pick 3 doses. A small, a medium, and a large, sort of, the not too big, not too small. And unfortunately, does that work in pediatrics? Not at all. Why not? In the first month of life, your metabolic enzymes in your liver change dramatically, dramatically, dramatically. So whatever we dose on the first day of life, the next day is on or off, or you have to give it twice as much. And we've seen this over and over again. So thinking that you're going to get a dose in adults and you're going to what we call weight or age de-escalate it down to a child is virtually impossible. So they do an adult phase 1 study. They think they get a dose. How often do they pick it right? We'll talk about that data in a minute. Then they go ahead and do a phase 2 study. A phase 2 study is a little bit on safety. What's the problem? How often is a safety signal in 100 patients? If it's 1 in 100, you enroll 100 patients on your open label safety study, you'll see maybe one event. What are the error bars around that? Anything from -2 to +10. So you could see none and think it's safe, or you could see 10 and think, oh my God, it's not safe at all, because those are real error bars. So thinking you're enrolling 100 patients and getting a safety signal or not getting a safety signal is completely, prone to guess. Too often, I read all of the news from all the pharma companies. They will do a big blast. Phase 2 study showed efficacy. Can a phase 2 study ever show efficacy? No, it's not sample size to show efficacy. It's show safety on a 1 to 100 level. So number one, you can't say anything about efficacy. And number two, you probably can't say a whole lot about safety either. But if you're someone like me that looks at the stocks and looks at the news, time after time again, you'll see the phase 2 study blasted all over the place, great, great, great news. And then it comes to the phase 3 and it's like, because the phase 3 failed, because the phase 2 only had 100 patients in it. Then you move on to the phase 3 study, you've got a couple of thousands in there. Then you have to go ahead and do review. You have to pull all the data, and we're going to talk about that review step. Why? Because that's the perfect place for AI to start going into our clinical trials, right? You've done 1,000 patients, randomized compared to standard of care or placebo, and now you have to actually look at the data. The issue is the EMA likes its tables this way, the FDA likes its tables this way. So you have to take the exact same data sets and manipulate them for the different regulatory bodies. And then, in my situation, oftentimes the regulatory bodies don't agree. The FDA wants a weight band of 2 to 6 years of age. The EMA wants a weight band of 1 to 5 years of age. So that means you have to take the entire data set and manipulate it all over again, and you can imagine that's a 6-month endeavor using SAS. We don't want to use SAS anymore. We want to use AI tools. So go ahead, they do it, they do the new drug application, they're approved and all the other stuff, and then they finally get,
Sharon,
yes?
What about the rare disease?
Orphan diseases. So orphan diseases in the US are a completely different bundle of questions because the rules for FDA and EMA don't apply. In order to get a licensure for an orphan disease, you don't go through the same regulatory pathway at all. You never do a phase 1 and a phase 2 and a phase 3. Why not? A, you don't have enough patients, and B, it's not ethical. So they will use a phase 1 to say, okay, we didn't kill anybody, or even if we did, we only killed 1 or 2, or they were going to die anyway, but we may have prolonged life in the few that are left. So that sounds like a very fancy idea. What's interesting is that in the US, TB or tuberculosis is considered an orphan disease.
Wow.
So if you look around at the rest of the world, tuberculosis is not so much an orphan disease. But in the US, TB is an orphan disease. So there are no studies that go in the US to look at multi-drug resistant tuberculosis. So if you have a good drug and you're a company, you'll go do the happy dance. I don't have to do a large phase 3 clinical trial to get it licensed. I could just do some safety and PK and say, call it a day, I'm done. Unfortunately, from that experience, we've learned a disaster. So they will get a licensure, say, look, the drug is safe, it's effective, it's great, move it to the international setting, and it fails completely. Sometimes from safety, even things like grade 1 or 2, you threw up every day. Well, if you had TB and you threw up every day with that new drug, are you going to take it next week? No. Or efficacy, it didn't work because you actually didn't have enough patients with multi-drug resistant TB to actually test it on. So depending on the orphan disease, you can get stuck in this black hole of you think it's a good idea to go ahead and not have to test it, but at the end of the day, you really needed to have tested it. So depending on the orphan disease, you get different answers from the FDA as to what to do. The other thing is the FDA doesn't like that you did the entire study in sub-Saharan Africa and you didn't do it in US because the populations are different, right? And what we have in the US, you can come into the clinic, I could see you every day, I can call you on the phone, I could see you taking your meds. I can't do it in sub-Saharan Africa. I'm lucky you come to clinic, I give you a bottle of meds, take it, okay, I'll see you next month. So the things that you want to compare to show efficacy, I gave you the med, you took the med, you swallowed the med, I can't do in a lot of countries compared to what I do here. So the FDA is wrestling with how to do it, and this change in leadership in the FDA is only making it worse. I don't know if you looked at the news today, but the FDA refused to look at Moderna's flu vaccine. And you sat there and you went, last year, you thought it was a good idea. We need a universal flu vaccine. And their complaint from the FDA was that they didn't use the best clinical comparator. And you sit there going, you're right, because we don't have a best vaccine to compare it to. We just have the standard flu vaccine that changes every year. So in order to get enough patients in, you use last year's flu vaccine for the population last year. Well, the FDA thought that wasn't good enough. I don't know what they wanted to compare it to, but now the fact that they're not looking at, which was a great clinical trial, well-numbered, well-positioned, no other vaccine manufacturer for flu is going to touch their toe into the flu shot now, because if they didn't like this, they're not going to like mine either. So we're seeing more of a problem with the FDA, not less.
And EMA?
So EMA is interesting because their reviews don't always have the same people. So a lot of times we'll submit something to the EMA, and they'll say, okay, great, we've done a pre-review, go ahead and do the study. And then we'll come back with the study, and they're like, well, we told you a weight band of 2 to 8 kilos, but you didn't enroll enough 2 to 4 kilo kids, so go out and find more. No, the weight band was 2 to 8 kilos. We enrolled the 10 kids. Granted, half of them were 5 to 8, not 2 to 5. But to find a kid at 2 kilos who has a multi-drug resistant TB, you know, I'm looking for the diamond on the floor here. I'm never going to find it. So we're wrestling with that now because that's a while, on paper, they're right. You need to find some 2 to 4 kilo kids. But in real life, I would wait another 10 years to find enough 2 to 4 kilo kids to do a PK study on, because they have multi-drug resistant TB, to wait to get enough data to move to the next phase. So depending who you talk to, and even though EMA and FDA said, oh, we're going to look at what they said and we'll talk to each other and we'll do this together, they don't always see eye to eye on stuff. And that becomes a big problem as well. As you know, EMA moved out of the UK and moved into, huh? No, not Brussels. They moved to Amsterdam. So all of a sudden, the people who were doing all the work, none of those guys moved, so they had to reinvent everything all over again. And that becomes, you know, UK is not the EU anymore, and everybody makes their own rules and stuff, so it's always fascinating. So this is a sort of a bigger look at it in sort of in one place. You've got the discovery, preclinical, phase 0, phase 1A, 1B, really much more safety stuff, and then 2A, 2B. 2B is thought to be efficacy, but it's really, really a numbers game, because efficacy is dependent on what the efficacy objective is. Is it cure of disease, or is it their symptoms got better? Symptoms got better, you might need a whole lot of different numbers. Cure of disease means if you had tuberculosis, I treated you for 6 months, now I have to follow you for 2 years to see that you're actually cured. And if you relapse, you have TB again, is it that I didn't cure the first time, or you got a new infection the second time? So there's a lot of, I want to say, negotiating when you talk about an efficacy 2B disease. You've got your phase 3 clinical study, and I have here patients 300 to 3,000. So let me give you an example. When rotavirus vaccines first came out, the original Rotarix, that was a Wyeth product, was only studied in about 20,000 kids, randomized clinical trial 1-to-1, and they had a few cases of intussusception, where the colon telescopes on itself, and everyone got all wigged out about it and said, you can't have that vaccine anymore. It's higher numbers than would have been in the background population. 2 other drug companies stepped up and said, we have a better rotavirus vaccine. We will look at this. How many patients did they have to enroll on those? Any guess? The original Wyeth one was 20,000. Any idea how big the other 2 company studies were? Take a guess. Guess. 80,000. 80, 0, 000. 80,000 children had to be enrolled on that study in order to prove that the vaccine was better than the background rates of intussusception in the kids that got placebo. Wow. The other company only did 66,000. Okay, so just think of those numbers, and if you're ever doing a clinical trial, just think of how hard it is to enroll 80,000 children. Right, you got to get them to get their first dose before 2 months of age. So the second they're born, you got to find those mothers, approach them, consent them. They have to come back, get the 3 doses of vaccine before the 6 months, and then follow them for a year. Just think of the numbers and the cost of it. So, you know, I sit there sometimes and I look at this going, yeah, actually the numbers are often a lot higher. So I wanted to also just sort of sit there and look at sort of what FDA does, but more importantly, I wanted to show you the cost. And this is numbers that are probably an underestimate of the cost. These are a little bit old numbers, but they're as recent as I could find because a lot of companies don't want to share the cost of how much a study is because they don't want the stockholders sitting on this side going, what the hell were you thinking? Oh my God, that's ridiculous. So here is it. Phase 1, per patient is about $16,000, $17,000. A phase 2 for the average cost is about $1 million, you know, is in US dollars is about, you know, $1.1 million. That was only 50 patients on the study. All right, so you go ahead and tell your Kupat Cholim, tell your government, whatever, saying, I'm going to do a study. I only need 50 patients, but it's about $2 million. And they're going to go, no problem, you got it. Phase 2 per patient is about $18,000. The average cost in 2020 was about $2 million. A phase 3, the average cost per patient is $27,000. And what is the average cost of the trial? $22 million. So you really, really, really want to be sure that when you wrote your phase 2 and you got a result, that you could, with a good face, turn to everybody and say, give me the $22 million so I can get the phase 3. So a lot of times, companies will look at this and go, it's not worth it. That gets into your orphan drug question. They're not going to spend $22 million to get a drug licensed for 10 kids. It's not worth it. On the other hand, they still want you to use the drug, and that's that interplay between, is it an orphan drug? Is the FDA going to approve? But then the insurance company go, well, you didn't do the phase 3, so we're not going to pay for it. You have to pay that $1 million per dose for that patient. Yes?
Can you explain why is it more expensive per person when you're saying phase 3 and you have more people?
Certainly.
I would assume it would.
No, so the answer is, it's a question of how long you have to do the study to prove that it is efficacious, because phase 3 at the end of the day is an efficacy study. So for the TB example, you're going to be on drug for 6 months. I'm going to have to follow you for 2 months. Yeah. For some of the other drugs that we do in the HIV realm, you may have to follow them even longer. Or any kind of HIV cure study, I'll have to follow you for 5 years. So that adds up very, very quickly. The other thing is, what is the standard of care that you're comparing it to? If it's placebo, oh, that's cheap. But if you're comparing the new cholesterol med to the old cholesterol med, are you buying it? How are you getting it? And how expensive it is? And then you have to also monitor that for safety. So the numbers get much, much more complicated the bigger the study, and you need much more people to watch those patients. If I'm only watching 5 patients, it's part of a nurse and I'm done. If I'm watching 25 patients, I need much more staff to actually do it. And then much more data equals much more data entry people equals much more statistical analysis before I get to the report. So it's not the per patient that's so easy and cheap. It's the cloud around them that starts. So when we cost out a clinical trial for us to do, the first thing I do and when I develop my budget is I say I need $800,000 for the behind the scenes work. The statistical guys, the data management guys, the staff guys, the training, building the CRFs. That's money that someone has to put in before I even got to giving the drug to the kid the first time. In addition, that $800,000, I also have to make drugs specific for that study, because the drug I'm making for that study is a single lot. And It's quite different when I have to massively produce drug for everyone on the study. So there's a lot of back and forth things that all of a sudden start adding up, but people don't realize it because they think, oh, it's just someone sitting there at the desk and typing the clinical trial. Oh, you have it. Why did it take so long? No, it wasn't me sitting at the desk. It was the cloud around me doing the background work. Does that answer the question? Yes? So phase 4 is really fascinating because each company is required to do a phase 4, but there's no time limit on the phase 4. Because you have to use phase 4 means the drug is licensed, and now you have to do big picture, how is everybody doing with that drug? If it's a drug they're taking up easily, you can get the data easily. You can use EMR data, and other kind of data, so it's pharmacy data. They took it, hospitalization data, that they get sick. But it requires taking up the drug. So now you have a new GLP-1 drug. All right, great, new GLP-1, you want to do the phase 4. How expensive is that drug? If it's going to be 20 times more expensive than the drug they're on, no one's going to take it. So thinking you're going to do that phase 4 quickly, you can't. So then you start advertising on television, the new GLP drug, you take it by mouth once a day, you tap dance around the stage and stuff like that. Everybody's swinging and dancing and stuff. No one's taking it because that switch from their generic now, injectable, to the oral brand name isn't going to happen. So phase 4s can last forever and ever. We have suggested on the pregnancy side that we want registries. Instead of saying to, FDA saying to a company, do the phase 4, we want them to say, develop a registry. So that if someone is given the prescription for that drug, that doctor, whoever, can enter that patient on the registry and then we could automatically follow it. Otherwise, you're at the whim of the pharma company to follow the phase 4.
Don't you think that currently, when we have so much data in EMRs, we can do the phase 4 regardless of what?
So that's what we're trying to do. The problem is most of the EMR stuff in the US is disconnected the hospital from the general physician. So the hospital drug data I can easily get, and I get it all the time. But if you go out to your private practice doc, and that private practice doc is on a different EMR system or on no EMR system or a bad one, and it's not linked to their private, or it's linked to a different system, you lose them. So I'm going to talk about that in a few minutes of how they're trying to do that. But you're right, it should be easy because a drug name is a code. You should be able to pick up the code.
Yes, this is what we have suggested to the Ministry of Health in Israel because we have inpatient and outpatient in one EMR.
Is one linked. We don't have that. So most people in the US use either Cerner or Epic. There are a few other smaller ones. But the problem is the coding from outpatient doesn't exist the same way that the coding on the inpatient exists. And that's been our big problem because we can't track it on the outpatient. Countries, Denmark, for example, that's also just one system. It doesn't matter where you're standing in Denmark or who you're seeing, your same medical record number. In South Africa, we found that every time you went to the doctor or the hospital, they gave you a different EMR, even within the same hospital. So we couldn't track the patients just in that hospital alone because they had different EMR numbers every time they came. At our hospital, we found a glitch in the system. When a woman would come and deliver, the baby would be baby girl Nachman. But then when they came to the clinic, they used the married name. So there was no link between the two names because they didn't link them up at all. They didn't say, oh yeah, it used to be baby Nachman, now it's got a, no. Those two names never collided. So when we would see the kids, sometimes we look at the mother going, you look familiar. And you know, sometimes I think, oh my God, I'm being stupid, telling a mother, you look familiar. So these phase 4s are a very big problem for us. So although pharma and biotech companies have continually tried to increase research and development investment, the number of new drugs gaining regulatory approval per billion USD dollars spent has halved approximately every 9 years. So we're spending much, much, much more money and getting much less out. And one of the big problems is the high failure rate of clinical trials. Less than 1/3 of all phase 2 compounds advance to phase 3, and more than 1/3 of all phase 3 compounds fail to advance to approval, just like the Moderna one. Phase 3, everything was good, not going anywhere. because of these critical checkpoints don't occur far into the second half of that R&D cycle, with the most complex trials carrying 60% of the overall costs, most of the companies actually don't want to do it. This becomes especially a problem for me in pediatrics because I think they should be doing these drugs in kids. So this is some data from one of the Nobel laureates in economics. And basically the bottom line is the return on investment in young children actually outweighs the return on investment in adults. Why is that? Because prenatal and postnatal health sets a lifelong trajectory of health and disease. So if you fix it early, you're not going to see it late. You know, when I see all these obese adults, I sit there and go, so what did they look like when they were a kid? And you ask them and they had obesity their entire life. Well, if I had fixed it when they were a kid, I wouldn't necessarily, I might still see some of it, but I wouldn't see the volumes of it that we see in adults. Now ask me, are any of the GLP-1s approved in pediatrics? Yeah, no, they're not. So that whole, dilemma isn't getting fixed because you're not hearing where the problem started. You're starting to giving Lipitor to adults is very nice. But if I looked at the cholesterol in the kids earlier on and I tried to fix it then, maybe I wouldn't have so many heart attacks in adults. Of course, if you're Ashkenazi Jewish, you have high cholesterol anyhow. Sorry, it doesn't matter. Sorry. So when we think about clinical trials in children, we don't think of children as unique characteristics. We think of it as the first 1,000 days of life sets that lifelong trajectory for health and wellness. That includes the mother, the fetus, and the infant for those first 1,000 days. Because if I detect the problem then and I fix it then, I'm not going to have the problem later on. This becomes an issue for us because when we approach pharma to say, we want to study infants or children, I get the, oh my God, hair on fire kind of approach because they're happy to study 12 to 18-year-olds. But the dose in a 12 to 18-year-olds is almost identical. I can't think of any real exceptions to adults in adulthood. So telling me you're going to age de-escalate into adolescence like you've done such a great job and given me a fantastic boon doesn't help us at all because it's the exact same dose. So we went back and looked at all the drugs that were approved in the past 10 years in adults, and then we looked when they were approved in children, and then we looked at the package insert to see, was a new toxicity found in a child that didn't get found in an adult? And what do you think the answer is? Zero. There were new toxicities in adults that we didn't see in kids, but there was never a toxicity in a kid that was novel that wasn't already seen in an adult. So this idea that I'm going to find a new toxicity that's going to throw my drug in phase 2 or phase 3 out the window actually has no basis in reality. So that's why we really keep pushing and pushing and pushing that we shouldn't protect children from research. We should protect them in research. And that's that ethical obligation that we talk about all the time.
Sharon, what about the COVID vaccine?
So we studied COVID vaccines in kids quite easily, and we had lots and lots of families sign up. We did both the Moderna and the Pfizer vaccine at my institution, in different cycles kind of a thing, and we had people throwing themselves in the hallway to get on vaccine studies.
Because in Israel they found toxicities that were specific to young adult males.
Right, so the question is interesting. So what's the toxicity? The question was that perhaps in more adult males, and maybe there were some females also, they had a cardiac toxicity from the vaccine. So the answer is, in the snapshot of studies that were done, yes, that was true. But the question is, was it the vaccine? Was it an immune response to the vaccine? So we took 3 steps back and looked at the cardiac toxicities of the illness. So when we compare the cardiac toxicities of the illness, they also had the cardiomyopathies, but they often wound up dying from it or having long-time adverse events versus the cardiometabolic toxicities from the vaccine. You treated them with some Advil, they got better, and there was no lifelong consequences. So it looks like it's an immunologic response to the protein that was in the vaccine, which is also the same protein that's in the virus. So that philosophical idea, I gave the vaccine, I was going to cause harm, the answer is actually, if you gave the virus, you'd have a lot more harm because you would have done the exact same thing. So it's interesting in that since then, we've still given out lots of vaccine, but we've actually never seen any newer numbers on that, cardiometabolic stuff anymore after that. So I really, really don't know where it was. Was it that vaccine and that virus that circulated at that time? Or was it something else? But it doesn't look that it was vaccine related, but rather, I want to say
Immune.
some immune related, some piece of it. And because we know what the part of the virus is that went into the vaccine, you could kind of center where it was, because that piece is always in the virus. It's not ever turned off. So it's interesting people got crazy about it, but because it was a numbers game, the incidence of 2 doesn't mean anything when you look at 100 versus incidence of 2 in 4 patients. So it's too bad it came out that way, but the bigger numbers don't support it. Okay, we also think about what are the physiologic changes across the life cycle. So there's a lot of hormonal and anatomic changes, pregnancy, postpartum. Of course, anyone who has a teenager, we know there's hormonal changes in that population. But I really wanted to remind everyone it's also the developing brain. And the developing brain of infants, children, and adolescents are very susceptible to neurobiologic effects of early life infections. If you give a kid herpes in their first year of life, you've squashed them for their entire life. You give them congenital CMV, even the asymptomatic kids don't look good later. So we just finished 3 years of our, and I'll be talking about this when I'm in Israel in the spring, we just finished 3 years of our congenital CMV long time follow-up. So we got a grant from NICHD to test every baby in New York State in 2022 for CMV using the heel stick blood PCR testing. We tested 210,000 babies, and we looked at who had CMV on that PCR. We went back and verified it by urine or saliva, depending on the institution, and then we prospectively followed those kids. The reason New York State was worried about CMV is they said, oh, it's a hearing loss thing. So they said right now they're screening everybody for hearing loss, and if you failed hearing testing at newborn, you got CMV testing, and that kind of didn't work. So we did it the other way. Let's get everyone CMV tested and see how many. So how many babies do you think had hearing loss at that first test from 210,000 babies that we tested? 11. Not worth it. So, but we did, more interesting is, amongst the asymptomatic kids, and we had a couple of hundred asymptomatic kids, we continued to follow them. And in the first year of life, we picked up another 8 that had hearing loss that didn't have it originally, which tells us that this idea that you think you know what you're going to find is completely wrong. So, we're wrestling with what to do with the kids now, and now as they age up, we're doing all the MCHAT testing, looking for autism triggers and all the other stuff, and we're hoping to follow them for at least 1 more year, but grant situation is always good these days. So, but it's interesting in that people always went, oh, hearing loss, if you didn't have it at birth, you're not going to have it. And the answer is that's not true. So those kind of infections, we have to do a better job on figuring out, and the brain, that developing brain, is bothered by them. CMV is a double-stranded DNA virus. You don't ever get rid of it. So what you don't see in that first year of life doesn't mean you won't see it later on. So what are some of the rules?
Can I ask?
Sure.
In the previous slide, you mentioned hormones and anatomical and development brain. What about developing the immune system per se?
So it's a great question, great topic. I didn't put it on the slides, but the answer is we know less than we think we know. And that's a real problem. Don't forget, the fetus and the mother immunologically are silent to each other, because if they weren't, you'd kill off every fetus.
This is exactly why I'm asking.
Right, so the answer is those babies are immune tolerant. Well, how do we know this? In the HIV cure agenda, we've gone on to give BNABs, broadly neutralizing antibodies to those babies. In the adult world, that generates antibodies, autoantibodies to those BNABs, and you can't necessarily use it. In pediatrics, we actually never see ADA, and it's because they're immune tolerant. The question, of course, comes up, well, you gave them that antibody, what happens 5 years down the line? Are they going to make an antibody to it? We see that in the Crohn's patients. The BNAbs you give them, 5 years later, they have autoantibodies. You can't ever recircle that treatment again. But what's interesting is that how immune tolerant those babies are, that we can give them those BNAbs on the HIV cure spectrum, trying to get to cure, cut down a reservoir, and they never make autoantibodies to them.
And interesting also in the aspect of breastfeeding, which could be 2 years or whatever, so antibodies are shared. I mean,
Right, but even amongst the breastfeeding, what's interesting is that both breasts are not the same.
yeah.
The macrophages are different,
Yeah.
free virus is different in the two breasts. You could transmit virus on this breast and not on this
Very
breast.
dynamic.
Yeah, so there's
situation that-
right, as I said, we know less than we think we know. Because every day we do something, we're like, huh, look at that. I didn't know that. Yeah, the two breast thing, when I say that to people, they look at me like, are you kidding? I'm not kidding. It's really that way. Okay, how are we doing time-wise? Yeah, we're running a little late. Okay, so what are some of the rules? Number one, you don't need to look for efficacy in kids. If it works in adults, you don't need efficacy in kids. Vulnerable populations, you're not allowed to say anymore, but those do require additional protections. Pregnant women, are they adults or are they pregnant? And every company says yes. I don't know what the answer is. Okay, basic ethics. Kids should not be enrolled in a clinical trial if the scientific or public health objectives cannot be met through enrolling subjects who would provide that. You have to absent a prospect of direct therapeutic care a lot of times in the phase 1s before you can enroll a kid. You have to say, is there benefit or no benefit? And children should not be placed at a disadvantage after being enrolled in a study, either through exposure to excessive risks or failing to get necessary help. And there are different pathways to pediatric licensure. You have sequential development, you have a parallel development, and then you have the product is being developed only for pediatric indication. So for example, the Hib vaccines, Haemophilus influenza type B, were never an adult product because no adult gets Hib disease, they can't. After age 5, you make your antibodies, I don't need to give it. So you could never have done that vaccine study in an adult, you could only have done it in a child. Okay, so now let's look at some of the gaps. So in AI, what's the value and whose hands is it? Is it in the investor's hands? Is it in the data repository? Is it in the startup? Is it in the health provider? Or is it in the patient? And we have to sit there and think about who is the recipient of that information. Are we allowed to go ahead and grab data from EMRs without patients allowing us to collect their data? Is their data going to be anonymized, not anonymized? How can we use it? And a lot of institutions now have a sort of a signature thing when you come in to say that your data could be used, but it would be anonymized, de-identified, et cetera. I read all what we call the DUAs, the data use agreements for the institution. Every institution wants to collaborate with another institution. You need a DUA. The DUA rules in the United States are very clear. Is it de-identified or is it anonymized? De-identified, if it's still got the patient's zip code in there and they have a rare illness, well, it's not de-identified anymore. So you really have to understand what the rules are in your country and what the rules are for sharing. So for example, sharing data from EU to the US is almost impossible because the EU is very strong on if something happens to that patient, you, whoever got the data, are still responsible for their medical care, for their lives, which kind of like, yeah, no, I'm not doing that. And it's similar in Canada also. So we are trying to figure out how to go around some of these rules so that data can be shared across institutions. So I thought I would talk a little bit now about what's the project management roadmap when we write a clinical trial, and where are the potential areas for AI involvement. Well, step one, the writing team is formed. They have to be identified, they have to get assignments based on the sections, and the initial sections, and I'm going to show you some of these, are the schema and the objectives, the study design, eligibility, meaning inclusion, exclusion criteria, and the schedule of evaluations. Then we go on to other sections, and each of these stop points is an area that we think of AI could be helpful. And we have to avoid the pitfalls of AI. We don't want AI to be writing things on background and referencing studies that don't necessarily exist. And we've all read about those kind of, things in the literature. They quoted a study that didn't exist. Then we have to have our reviews, and then we have to finalize the protocol. So let's look at some of the parts of the protocol. So this is one of our studies called 2044, and it's looking at safety and PK of ceftriaxone and benzathine penicillin during pregnancy. So you're going to say to me, why the hell are you doing it? Both of these drugs have been used for years. We know to use benzathine penicillin for syphilis. Why are you studying it? And ceftriaxone we use all the time during pregnancy. Why are you studying it? And the answer is syphilis. We want to develop a better ceftriaxone. And the only way to develop a long-acting ceftriaxone is actually to get the PKs in the non-long-acting and the benzathine penicillin, which is already long-acting, and look at the area under the curve. Look at how to make a long-acting so we can take our current ceftriaxone, which is generic, and nobody wants to touch it, and build a long-acting ceftriaxone so that then when we have those patients who come in with their syphilis, one shot and you're done, just like we do with the penicillin. Why am I bothering? Because as you know, we're running out of benzathine penicillin. I don't know what the situation like is in other countries, but as you know, the byproducts to make benzathine penicillin are toxic. And that's why no pharma company wants to invest any money to build a plant to make benzathine penicillin, because they don't want the toxicity of the byproducts to make it. And it's generic, so they're not going to make any money. So the only way to develop a better drug for syphilis is to think out of the box, learn the PKs of the ceftriaxone across the different gestational ages, take that information, what we know about how to build a long-acting, now we know how to build long-acting, we're smarter, we got the nanotechnology, build the long-acting ceftriaxone, put it back into women, and then look at the PKs afterwards. So it's not a short-term problem answer, it's a much longer look at what we have to do. Anyhow, so this is the first step of the study. And as you can see from the schema, it's got the purpose, it's got the design, it's a phase 4, right? Because these are licensed drugs. It's got the population, it's got the sample size, up to 78 women for 60 evaluable, because we know that you enroll 60 women, not everybody will be evaluated, because we need all those PKs at all those different time points. And it talked about gestation age and the study duration. The next are, what are the protocol objectives? Why is that important? Because that's what drives the sample size. And if you don't write clear protocol objectives, that's when in the phase 2, you write crappy protocol objectives, and you say, I was successful, you fail in the phase 3, because the phase 3 objectives, phase 2 primary objectives are what drive the sample size. The secondary objectives are just your best guess. Oh, we're gonna look at safety, and we're gonna look at are they happy, and did they come back? Those mean nothing. Those are secondary. So when you read a study in the literature that talks about efficacy in a phase 2, you can't, because efficacy would have been a secondary and not the primary, not sample sized for that study. And then of course you've got this exploratory stuff. Did they like it? Were they happy with it? How did the kids do? So here's sort of an overview of the study. And this is very, very clear in our protocol, and you can always look online and see it. It's all open access. And it's got arm 1A, 1B, then arm 2, and all the different time points when those laboratory evaluations will be. So if you look closely, you can see that there'll be at 0 hour, 2 hours, 24 hours, 4 days, 7 days. So really need those women to come back multiple times, and they're pregnant. And pregnant women usually have another kid at home. So we have to work very hard with the women to make sure that we can either bring them in, send a car service to pick them up, or pay for the car service or something like that. Otherwise, we're going to lose them. Questions? Okay. So now these are the inclusion-exclusion criteria. And as you can imagine, inclusion-exclusion criteria become very, very, very complicated. And if you don't spell out exactly who you can come in and what they can come, you won't get a good study. And this is just sort of the schedule of what the protocol looks like. And every single one of these sections, drugs under study, study visits, et cetera, is available for us to use AI for. Because right now, we talk about it, and then we write it, and we correct it, and we write it, and we correct it. And then someone new from the pharma company will come in and go, wait, I didn't like that you said that. And you're like, we discussed this 6 months ago, don't you remember? But the use of AI now has the ability to summarize our prior discussions and say, section 4, 4.2, here's what we said, we're not revisiting it. So we're already using it a little bit, but we want to use it to write the sections ahead of time as opposed to retrospective saying what we did. And of course, and here's our scheduled evaluations for the study, and you can see why is this important? because this is the data that's going to drive the regulatory submission. So if you don't spell out exactly what you want here, and then use the AI tools to make sure that the tables, figures, and legends match the data here, you're stuck using SAS. And I don't have to tell you, SAS is, so last century, as my children say. We don't really want to use it. So beyond protocol, what are the next steps towards the licensure pathway? Well, there's study oversight. There's interim analysis, and I want to focus on that for a minute, because that has the report generations and all the memos to protocol teams and updates. And we want to be able to use AI for that as opposed to people. Why? Because people go home at night. And you work a full day, and you're tired, and you go home, you make mistakes, etc. But if I can use AI to derive those reports, I can have people checking the reports, but I don't have to have them start from all over. It's the same idea with radiology now. Radiology is a field of medicine that is dying quickly. People aren't going to go into it anymore. Because at the end of the day, you get tired, you make mistakes, but the AI reader for chest x-rays doesn't ever get tired, and they can go through 100 chest x-rays in an hour where you really can't. You do need someone to back check it, but the reading ability for chest x-rays now by AI has surpassed physician's ability, and the same is true with CAT scans. So there are things that we're already doing better that we need to do better in clinical trials as well. Then the trial master files. There's something called an ETMF, or an electronic trial master file. And what that does is it tells you, after you have version 1, everything that happened. The FDA takes that electronic trial master file and they check, how did you make a decision? So if you said this kid came in with vomiting, or they came with a rash, and I said, that rash, oh, it's chickenpox. It's not related to study drug, not related. They want to know who said it was chickenpox. And did you prove it was chickenpox? Or did you just do wink, wink? It's a viral rash. It has nothing to do with the drug. Well, in fact, it was clearly related to the drug. So the electronic trial master file records all those decisions and tracks them, who said what to wear, what to call, et cetera. So when they go back and look at the data and they see rash and it's not related, they know exactly where it happened. So we want all of that to also be available for AI for summarizing data so that someone can pull back and say how many rashes happened on day 1, day 5, day 7. And then, of course, there's that regulatory submission, and that's a big thing. When I did the raltegravir was the first integrase inhibitor we used in HIV for children, so I was the PI of that study. I had a lot more gray hair. And the file that went to the FDA, I think, was 100,000 pages. And you just think of it, and you were like, how many trees did we have to kill for this? And nowadays, here it is on a data stick. Okay, you're done, no big deal. But the volume of stuff that we had to write, it was the first time integrases went into children. We had to do all the PKs, all the safety, every adverse event, explain every single thing, was just hours and hours and hours and hours of work that we don't want to be having to do that. And we want this all automated, AI questioned, et cetera. So what are the other documents? Well, how can we use AI to improve speed of developing study materials, right? You come in, you have a patient, you want to put kids on a new TB drug, just sitting the mom telling them, your kid has TB, we're going to start a new med. They're going to go, no, thank you very much, give me the old med I don't want to be experimented on. I don't want my kid to experiment. You have to develop study materials, so we want to use AI tools as well to help us to develop study materials. And we have to do study implementation materials, lab processing, all the other stuff. So I'm running a little tight on time here. Just go through some of the FDA EMA stuff. In January of 26, the EMA and the FDA published a joint set of guiding principles for the good AI practice in drug development. The principles setting out a shared regulatory vision for using AI across the entire development of a clinical trial. This collaborative initiative aims to maximize AI potential to be able to use it across the world. So, and what are the things they want to put in there? It's human-centric by design. It's a risk-based approach, adheres to certain standards, clear context of use. It's multidisciplinary, risk-based. All this stuff is part of it. Do I think that people will be using it? I don't know. It's out there for the first time, so they've all agreed that we all need to think about it. And it's interesting, since I deal with a lot of pharma companies, they always say, the network is too expensive. I can go to a CRO, a research organization, and I could have them do it for cheaper. And I'm like, all right, go ahead. let me know how that goes. Then they'll come back and go, oh, they were so expensive. Most of the CROs want $1 million on the table now, up front, before they'll even talk to a pharma company for any study. Just cash on the table before they start. We're cheaper than that. So, who knows what's going to happen with a lot of this. So I'm going to stop there. We've got 10 minutes left. Tell me what kind of questions you have. I wanted to talk just for a few minutes about EMRs. And let me actually see if I can find those slides for you, because we're doing that now. So there are several networks across the US that are only using EMRs for data. The first of these is called PCORnet. PCORnet is federally funded, and it's got like 7 different studies and a site, groups across the country. And basically, they want you to use the EMR data. In New York, we belong to a group called Insight. Insight includes Columbia, NYU, Cornell, Sinai, Montefiore, and Stony Brook. And we take all of our data using OMOP data sets to collapse it together. It's obviously de-identified because it's gazillions of pieces of data in there to actually look at problems. As an example, one of the things we looked at recently was the autism stuff. I know we had great lectures yesterday on autism, so we're not as sophisticated, as advanced at that. But we looked at autism rates over time, and then more importantly, what happened when they turned 18? To see, are they getting the same high quality medical care that they were getting as adults? And the answer was no. So that paper we're writing up now, because you had the autism diagnosis, you followed the kid, you could see how many times they went to the general pediatrician, the endocrinologist, the neurologist, and they turned 18 and it extinguished. It was gone. Gone. And these systems are all linked together on their EMRs. So we could actually track the kid if they were followed at Sinai and then they went to Columbia or to NYU, even though those hospitals are independent, because we could geoscape where they were and found that the rates of follow-up and diagnoses and care was so abysmal that we're clearly missing the boat. Sorry.
Sharon, is it only on autism or is it on any chronic disease? I think that any chronic disease has been much more followed.
Right, I think chronic diseases are a problem, and it's true for cystic fibrosis. Cystic fibrosis did it differently. Cystic fibrosis years ago brought med-peds doctors into the pediatric fold. So they started seeing the kids when the kids were young, and as they aged, they stayed in the CF center because they had adult providers there for them. So there was no loss of continuity. The autism stuff, when you talk to an adult colleague, I'm going to send you this kid with autism, nonverbal, they're like, don't send him to me, send him someplace else. I don't have time for it, I don't want to be bothered. So we've lost that ability. In our cancer center, it's the same problem. Some of the kids, when they age up and we still want to follow them, we have to argue with the insurance company that even though the kid is over 21, they're a long-term survivor or a childhood leukemia and lymphoma, we still want to follow them. And the answer is, oh no, they're over 21, they have to go to adults. The adults look at these kids and like, yeah, who are you and what are you doing here? Mm-hmm. We had this problem with our SCID patients. Our SCID patients, severe combined immune deficiency, we would save them. We would transplant them at birth, soon thereafter, blah, 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 whole 9 yards. Then they turn, 18, 19, 20, and the adult providers don't even know what SCID is, never mind what to follow. So I am still following a 37-year-old, that I've followed since birth. He's getting a big pig. He sits in the seats like this. But because the adult providers have no idea what to do. And I talk about the immunologic, this kid is on long-term IVIG, I can't, vaccinate, I have to worry about it. And of course now he has a wife and the wife has kids. We have DeGeorges like that. What happens with those kids? The adult providers will not touch them. So, not good, as we say.
And for the family, it's a nightmare.
Huh?
For the families, it's a nightmare.
It's horrible because they're used to us hugging those children.
Yeah.
Oh, it's their birthday, here's a present, you came back, graduation. I wrote college letters for these kids to go to college, and I'm still their pediatrician.
So I think we have to applaud Sharon. That's so you had some questions, but are there other questions? Yes.
Because I think on
Yeah.
maybe